Good morning to everyone. It's great to see all of you here. I think this meeting really gave us a chance to meet each other finally after exchanging many emails throughout the world. So I would also like to say that I was informed that we are a little bit ahead of time, which means I can talk a little bit more, which works with my slides. In order to understand why I like to talk is that I'm fully trained by Dan Kastner. I've been working with him for 25 years. Everybody knows that he likes to talk. So after working with him for so many years, I got to talk even in a language which is not my native language. So anyhow, so besides telling you the DADA2 story, I would also like to tell you a little bit about genetic studies and what we are doing with all these patients with rare diseases nowadays. So as I said, I've been working on genetics of autoinflammatory diseases for about 25 years. The first project that I ever worked on was the positional cloning of the gene which is mutated in patients with familial Mediterranean fever, which took 7 years, maybe plus, if you count all the years that then spent collecting these samples in Israel, right? But 7 years of working on a single gene, and I think that there are probably 50 people in this consortium, which finally led to identification of the gene in 1997. What is happening nowadays is really that we can find the gene or genes in a very short time. Sometimes it's just a matter of months, and it takes probably 6 to 12 months to do functional analysis, but altogether it takes about 1 year to find the gene, to do functional analysis, and to prove that this is a new disease-causing gene. So why is this all happening? So I will say 2 reasons for that. The first reason is that I really don't think that we are any better understanding the human immunology and guessing the right candidate genes. It's really due to new technologies. So now we are in situation having access to new technologies such as next-gen sequencing, whether we talk about exome sequencing or genome sequencing, to find these genes really in a matter of days. The other thing which is very helpful in these genetic studies is also that we have access to these large publicly available databases of exomes, so that when we find some variants, we can immediately go and check the allele frequencies of these variants in different populations. And the third component, which is very important for studying patients with rare diseases, is really to have a network, because we often end up having a singleton cases. And even despite having this technology and having access to these publicly available databases, unless you have that second patient that can confirm the finding from the first patient, you are at loss. You just don't know where to go. So it is very important to have networking between all of us and to work together, because with singleton patients, we are not going to get that far ahead. So the story of DADA2 basically started with these 3 patients who were referred to our clinic because they all suffered from systemic inflammation, early onset systemic inflammation. So as you know, our clinic is known in the world for studying patients with systemic inflammation. But in addition to that, one of them, who was referred to us about 10 years ago, presented with some neurological features, which was kind of unusual because we never had any patient presenting with neurological features and basically due to recurrent strokes, as this was presented to us, and systemic inflammation. We couldn't do much with that patient at that time, really. Even if we had the good technologies and we did exome sequencing and everything, having singleton, again, you don't get that far because you end up with a very long list of candidate variants. So it was really critical for us to identify 2 additional patients, as shown here in this slide. So basically, these 3 patients presented with early onset systemic inflammation and neurological features, which essentially suggested that they are likely to have some mutations, whether it's going to be de novo mutation or we suspected compound heterozygous recessively inherited mutations in some gene, for the fact that all 3 of them came from the outbred American population. If you work with patients that come from founder populations, then you go after homozygous mutations, and that was the case with the Israeli study, where they had the advantage to have a founder population presenting with a very similar phenotype, and they expected to find a homozygous mutation. And that's what we do also when we work with patients that have some other diseases. So in addition, although we knew that all 3 of them presented with systemic inflammation and also with these neurological features, it was still a long shot. We were not sure whether we will find that these 3 patients have mutations in the same gene. Nevertheless, we did exome sequencing in these 3 trios, which means that we sequenced patients and their unaffected parents. And after filtering for rare variants, which means for variants that have frequency less than 1% in general population, and under the assumption for recessive inheritance, meaning that we expect these patients to have 2 different mutations in the same gene, we noted the first 2 patients share mutations in the same gene, namely CECR1 gene that encodes adenosine deaminase 2 protein. That was a total surprise to us. I mean, we really didn't know anything about ADA2. The only gleam of hope was that we learned that ADA2 is homologous to ADA1 protein, which was mutated with patients that have SCID, at least in some of them, which suggested that this protein has to do something with immune cells, and that was sort of, encouraging to us to think that maybe this is really a disease-causing gene in these patients. So the 3rd patient that I showed on my first slide actually was a more interesting case because we observed that this patient had 1 mutation, missense mutation in this gene that he inherited from his father. However, from the previous patient, you saw that parents are carriers for this variant. So we were not sure really whether this is just a coincidence that this patient has 1 missense mutation in this gene, or that this patient should have a 2nd mutation in this gene that we can't really detect by exome sequencing on standard Sanger sequencing. So I remember talking to our clinician, Deb Stone and Mandy Umbrella, and asking, are you really sure that this patient has the same disease as the other 2 patients? And they said yes. So that was a signal for us that we should really go and search for that 2nd mutation, which is not easy to do because this was obviously a cryptic mutation. So the first thing that we did was actually we just look at across the gene in this patient to see whether we observe lack of heterozygosity, meaning lack of heterozygous SNPs, which immediately indicated, because we observed lack of heterozygosity, that this patient may carry some genomic deletion in this gene or may have a mutation in the regulatory sequences, which is going to basically shut down the expression of the 2nd allele. So after doing some additional experiments using SNP arrays, actually we identified the 2nd mutation in these patients being this 28 kb genomic deletion that he inherited from his unaffected mother, and we also showed that another unaffected sibling in this family is also carrier for the same genomic deletion. So here are the first 9 patients that we presented in our publication in New England Journal of Medicine. So here are the first 3 families. In addition, what I would like to point out here is that you can see here by color coding that these patients also shared some of these mutations, which is, again, encouraging when you work with all these rare patients and you have a good candidate gene and then you find these rare variants and some of them are shared among affected patients. It's a good sign that we are on the right way. The other patients were identified through our collaboration, both at NIH. One of them was referred to Dr. Goldbach-Mansky. Another one we identify through collaboration with Dr. Sophie Hamilton and also Dr. Mario Abinoun. And then this patient was also very interesting because actually I found this patient by reading news in the morning because there was an article in the news saying that there is a patient described in North Carolina who is too young to have strokes. That was the only thing that was written in the title of this article, which made me actually go and read this article. I contacted the physician who was mentioned in that news article, and I told him that we are working on a new disease and we believe that maybe your patient has the same mutations or mutations in the same gene like the other 3 patients we identified thus far. So that patient came to NIH, I remember, on Sunday night, a couple of years ago. On Monday morning, we got his blood DNA sample. By Monday evening, we knew that he has 2 mutations in this gene. And to me, this was really like a cherry on the top. I mean, if I had any doubt in my mind before that this is not a disease-causing gene, I knew that this is the right thing, that we are on the top of the right gene. So I'm just telling you these stories because I think that they are really informative for all of you to understand how is and what does it take to work with all these rare diseases. So then through our collaborators in Israel, we also identified 3 other patients that met clinical criteria for polyarteritis nodosa, or PAN. And they were referred to us by Dr. Cezao Zen, who is here today in this room. So these 3 patients turn out to have the same homozygous mutation that was discussed also earlier today by Dr. Jakob Berkun. So here is the Israeli paper showing basically their families of Georgian Jewish ancestry who were diagnosed with PAN. Also, they reported 1 German family. In this family, there were 4 affected siblings, and they turn out to be compound heterozygous mutations, to carry compound heterozygous mutation in this gene, as you would expect with families that come from non-founder population. So obviously, you can see that this particular mutation is a founder mutation in Middle East, and the allele frequency of this mutation is quite high in Georgian Jewish population, estimated to be about 5%. It's a little bit lower in the Turkish population, and also in our cohort, we had a couple of patients from Pakistani ancestry who also happen to be homozygous for the same mutation, which suggests that probably this mutation is also present in the Pakistani population, but we don't know what is the frequency of this mutation. So what I would like to say here in this table is that DADA2 associated mutations are either novel, which means that they are not reported in any of these publicly available databases, or they are found at a low frequency, which is again consistent with the recessive inheritance and seeing these patients in outbred population. So this particular mutation, which is fairly common in patients with DADA2, is found at the low frequency in the general healthy controls, basically. And nevertheless, after finding that there are 2 carriers for this mutation in the NHLBI cohort of exome that at that time numbered about 6,500 exomes, we contacted actually the group who ran that project to find out, are these 2 carriers random carriers or they are related? And we learned that basically these 2 carriers are brothers or siblings who presented with late onset strokes at age of 70 or 80 or something like that, which immediately basically suggests that maybe having 1 mutation in this gene may predispose some people to develop some symptoms, maybe even to present with late onset strokes, in the presence of maybe susceptibility alleles in some other genes. So having 1 mutation in ADA2 and maybe some other susceptibility allele for which we don't know where they are, in what genes they are present, can really can make people more susceptible to present with some cardiovascular diseases or maybe even strokes. So we tried to answer this question by asking family members, in particular grandparents of the patients that we reported in our paper, to find out whether they have some symptoms of disease, whether it's cardiovascular or strokes and so forth. But once you get into this age group of 70 or 80, there are a lot of cardiovascular diseases going on, so it's really hard to answer that question. So the best possible way would be really if one could really have access to cohorts, large cohorts of patients that present with late onset strokes, which I'm sure they exist somewhere, and then do targeted sequencing of this gene to see whether basically we see presence of these mutations also in the cohort of patients with late onset strokes. So here is the family that is seen at NIH, and basically in this family there are 2 affected siblings, which are shown here. They have 2 mutations in this gene. What is interesting is that their mother suffered from lacunar stroke at the age of 45 and presents now with foot drop, and also there was evidence for the maternal grandmother that she had also biopsy-proven PAN. So again, raised the question whether maybe people that are heterozygous for mutations in this gene may present with some clinical symptoms. So we also had opportunity to check the frequency of these mutations in the cohort of patients that we received from Dr. Peter Merkel. Basically, these are the patients that presented with late onset PAN or polyarteritis nodosa. And in this cohort of 96 patients, we identified 6 carriers. 2 of them carry B allelic mutations in this gene, and 3 of them had only 1 mutation. However, we cannot really rule out the possibility of having second unusual cryptic type mutation in the same gene. We also have access to this cohort of patients that are diagnosed with other types of vasculititis, which are shown here, altogether over 1,000 samples. And we are going to do also targeted sequencing in this cohort of patients that are basically diagnosed with other types of vasculitis. So Few words about genetic screening for mutations in this gene. So this is not a large gene. CECR1 gene has 10 exons. It's encoded by 511 amino acids. And basically, I kind of like to work with these genes. Internal joke in our lab is if there is a candidate gene that has more than 10 exons, I just don't want to work with it. So this gene definitely met the criteria. It was a good candidate gene besides the fact that it has only 10 exons. Nevertheless, genetic screening for DADA2 is very, very complicated. And it's more complicated than what I see by screening other genes that are associated with autoinflammatory diseases. The reason for that is really that this gene is so rich with SNPs, or basically variants, nucleotide variants, that it really takes major effort for us to go through this and to be really sure that we are not calling SNP a disease-causing mutation and vice versa. So I always like I asked actually Dr. Ching Zhao, who is going to come here to this meeting later on, I always ask her to check my interpretation and vice versa, because I'm just always worried that maybe I will misinterpret one of these nucleotide variants. And so what you can see here in this table is, this is a table that I found on the exact database. So basically, it says, in essence, that this gene somehow tolerates a presence of a lot of nucleotide variants, as shown here by this constraint matrix depicted with z-score. So having a higher z-score, it means that that gene doesn't really tolerate very well presence of nucleotide variants. You can see that the z-score is very low for this particular gene. Likewise, also that this number, or metric score, PLI, number is 0. And again, it goes the same way. Higher number, it means that the gene is intolerant for the presence of loss of function mutations. And there are also 32 copy number variations reported around this gene. And this is something that haven't been explored thus far. We have to look into these copy number variations for the reason that we know that DADA2 is caused by loss of function mutations. Whether they are missense mutation or copy number variations, it doesn't matter. I mean, patients don't make a lot of ADA2 protein. And nobody really has looked into this, except that we published that one deletion, which is a 28 kb deletion. The reason is very simple. It's much harder to look for copy number variations than to do sequencing of this gene. So in terms of type of mutations, they are mostly missense. Today, there are more than 20 mutations reported as associated with deficiency of ADA2. There is one genomic deletion, as I mentioned. There is also one stop gain or nonsense mutation that was reported in a patient from Turkey. We also heard about splice site mutations. I'm just talking about one because the other one that were presented earlier today haven't been published. And again, there is this whole issue of cryptic mutations and how we are going to find these cryptic mutations. So this is the table that I took from the recent publication from Marco Gattorno's group and Roberta Corsi, who is the first author. Basically, they summarize nicely all the mutations associated with this disease that have been published thus far in the literature. So we know that there are more than 20 of them. And what you can see here is that they are just found all over the gene. So in some diseases, you can go just to particular exon or domain of the protein to find disease-causing mutation. Here, we have to screen the whole gene. And again, fortunately, the gene is not that big, so it's doable. But again, these mutations, they affect the catalytic domain of the protein, protein dimerization domain, interaction domain with other proteins, and so on. So now I would like to say a few words and give you an update on molecular diagnostic that has been performed at NIGRI post our publication. So basically, if I exclude the original 9 patients that were published in that manuscript, thus far we have screened about 120 patients. Some of them were seen at our NIH clinic. Some of them were mailed-in samples, basically referred to us for genetic screening, and we identified 40 mutation-positive patients. By that, I mean 33 patients that carry BL allelic or double mutation in this gene, and also we identified 7 patients that carry a single, either rare or novel mutation in this gene, predicted pathogenic, which means most likely they have DADA2 deficiency. It's just that we cannot find that second mutation by doing regular sequencing. So we did some search to identify the cryptic mutation. However, it proved to be very challenging. So how one can go about screening for the second disease-causing cryptic mutation is by doing long-range PCR, QRT-PCR. We have to go into regulatory sequences. Regulatory sequences are found basically in introns between the exons. We have to go 5', 3', and so on. It's a lot of work. So, you know, one way to do maybe more efficient screening would be really to do the targeted sequencing of the whole gene, which after all, it's not that big. It's about 25 kb. And also we are thinking about doing genome sequencing. And the reason why we are thinking of doing genome sequencing is that exome sequencing does not allow us to detect copy number variations. So apparently, we can find them only if we do genome sequencing, because there are some softwares developed, you know, to identify copy number variations from a genome sequence. So in these patients, we identified 16 mutations that have not been previously associated with DADA2. Again, they are just found all over the gene. And here, I will show the evidence for a null allele, or basically that cryptic mutation. So here, there are 3 siblings that were referred to us from Johns Hopkins. They carry one missense mutation in this gene. And again, if you sequence their DNA sample, we see this double hit or double peak here suggesting heterozygosity. However, when we sequence them, their cDNA sample, you see only single peak, which suggests again that they have something which is essentially shutting down the second allele in these patients. Here is another family, again, a very similar story. However, this family was interesting because there was an index case who presented with multiple strokes and 2 other siblings that presented with much milder disease symptoms, which again, we heard about that today. There is a lot of genotype-phenotype I would say not correlation, but actually there is no good correlation between the genotype and phenotype. And we failed to identify the second mutation in this family. The question was really whether these 2 patients should be treated because they had mild symptoms, whether they really have that disease, and we only could find 1 mutation in both of them. So we, again, did measure ADA2 activity in their serum samples, and we showed that both of them have low ADA2 activity. So essentially, it means that confirms their clinical diagnosis of DADA2 despite very mild symptoms. And also, we carried on with the haplotype analysis, which basically showed here that these 3 children, unfortunately, inherited the same haplotype from this parent, which carries this cryptic mutation. So we are in the process, again, of trying to identify the second mutation. So here, again, we measure the ADA2 activity, both in patients that have biallelic mutations in this gene and patients that carry only single mutation and hypothetical or cryptic mutation on the second allele. And again, you see that they all fall into the same category of people who have low ADA2 activity. So since the original publications of deficiency of ADA2 protein, there are a couple other reports that also identify mutations in this gene in patients that present with the Sneddon syndrome, with Castleman syndrome, severe anemia, and immunodeficiency. I'm not going to talk about that. But I will just point here to this family with the Sneddon syndrome. So what is the difference between Sneddon syndrome and patients that we have seen at NIH clinic is really that Sneddon syndrome typically is explained as a disease which basically happens later on in life, maybe in a second or third decade of life. Some of the patients present actually in middle age, around age of 50 or something like that. Often they have high phosphoantilipid antibodies. So it is in the spectrum, but nevertheless, we expected to find mutations. But nevertheless, still the question is why some of these patients present, you know, at age of 50, and some of them present very early in their life, despite the fact that they carry biallelic mutations in the same gene. There was also a report of the patient with the Castleman's disease. So this patient presented at early age of 5 years with recurrent fever, splenomegaly, lymphadenopathy, elevated acute phase reactants, and polyclonal hyperimmunoglobulinemia. So you remember this morning we are talking about hypoglobulinemia that we see typically in patients with deficiency of ADA2. So this patient basically had high serum levels of IL-6, also immunostaining of lymph node biopsy confirmed strong expression of IL-6. So clinically, he fits diagnosis of Castleman disease, and he responded to treatment with tocilizumab or IL-6 inhibitor, and he was reported to have the same mutation that was basically identified in these patients who presented with PAN. So I really don't know what to say, except that it could be that he is really not expressing symptoms of DADA2, or that he essentially has two sort of diseases, concomitant phenotype. In addition, I would like to say that I had the opportunity to test a couple other patients that were diagnosed with the Castleman disease, and I was not able to find mutations in any of them. So I think that what I would like to say here is that there is a lot of phenotypic variability even with patients that have the same genotype, and I think Joris is going to talk about that later on. And again, the phenotypic variability comes to their age of onset of disease. Some of them present with neurological involvement, some of them not. But what I found interesting in this paper is that the ADA2 activity, according to that study, tended to be lower in patients with stroke compared with patients without stroke. So basically, that suggests that there is a role for modifying allele in this disease, so that in addition to the genotype that we find in this gene, there is a role for other genes that are interacting with ADA2 and can modulate the expression of ADA2. Here is another example of phenotypic variability, but considering that we are short with the time, I will skip it. I think Paul Brogan discussed this family, which is interesting because there are also patients that with incomplete penetrance, so that they are perfectly fine. They have no symptoms despite having the genotype in this gene, something to keep in mind. And basically what I would like to say in conclusion is that rare pathogenic mutations in ADA2 are associated with the spectrum of phenotypes, as we have learned during the past couple of years. We started with fevers, vasculopathy, vasculitis, a little bit of immunodeficiency. Now we know that there are a number of patients that present with immunodeficiency and not much of vasculitis, severe anemia, which is a total surprise to all of us. So phenotypic variability also includes incomplete penetrance, and that's not unusual. There are many other human diseases where basically there is incomplete penetrance, whether they are dominantly or recessively inherited. And also I would like to point again for the role of disease modifiers. By that I mean other gene alleles or alleles in the other genes, and also possibly for the role of some epigenetic and environmental factors. And again, to say that there are cases with cryptic of null mutations complicate CECR1 genetic screening and highlight the importance of using multiple diagnostic measures. So we are often in a situation when we can't find really the second mutation. We run this ADA2 activity assay to confirm the patient's diagnosis, clinical diagnosis, which would then suggest that these patients should treat it as soon as possible with TNF-Inhibitors, because it's going to take a longer time to find the second cryptic mutation. So finally, I would like to make some acknowledgments, in particular to Dr. Jing Zhu. She also goes by the name of Pearl. Other collaborators from the NIH intramural program, in particular Dr. Daniel Kastner, and our collaborators throughout the world who help both with the functional studies and also by referring patients for this study, and of course to DADA2 Foundation. Thank you very much for your attention.